This was a very exciting session, I thought, because we covered three areas that I believe are very important at the moment in connecting our understanding of the basic biology of Parkinson's disease and what can be offered and done for the patients in a clinical setting. So this is why this was a translational session. And here we had three experts covering three topics that are very hot at the moment. One is on lysosomal dysfunction can be used not only to understand the basic biology, but also eventually as a possible target for intervention. And here, Dr. Leonidas Stefanis from Greece discussed the potential of lysosome-targeted therapies in Parkinson's disease. So lysosomes are these organelles that exist inside cells that basically degrade damaged and aged proteins as they accumulate. And we know this is a central hallmark of Parkinson's disease. So if we can understand how we can help the cell deal with the accumulation of damaged proteins, we think this might be beneficial and that this might have therapeutic potential. Then afterwards, we had Dr. Malou Tanzi from the US talk about the immune system as a target for intervention in Parkinson's disease. And here, Dr. Tanzi discussed the role of the immune system as a target. And this is based on very new data that is implicating our own immune system in various responses that are present during Parkinson's disease. So we know that because we produce these proteins that accumulate, they may end up circulating in our bloodstream. Our body tries to react and respond. There are neuroinflammatory processes that are perhaps even inflammatory processes in the periphery. There's the involvement of immune cells that will try to go and help our body deal with the problems that arise. So again, if we find ways of helping the immune system deal with the problems, this could have tremendous implications. And finally, we had Dr. Eliezer Maslia, also from the US, talk about antibody-based therapies. And basically what he did was to summarize the status of the antibody-based approaches. And also he told us about the work that led to the current stage of the research and of the trials that are ongoing. So this is really one of those examples where we went from basic science, from basic ideas we had in the laboratory to something that can be and that is being tried already in the patient. So it's very exciting, this research. There are promising results already ongoing. These were presented in another session, so we will cover this in a separate question. But I think this was overall a very exciting session because it really told us about how laboratory research can be translated into clinical applications.